Hello? Oh. It works, yeah. So, I think that when you have a molecular signature together with unique morphology, it may convince you that you have a new species. But it may happen that the single specimen you have found is not characteristic enough to describe it. For example, once we have described from Australia, Alvisia repens, based on peculiar morphology and unique signature, molecular signature. After years, we have found that, yes, this is a good species, but it is not reptant. So the epithet we used and the character we consider to be as the most important is wrong. So this was an aberration. Species is correct. It really exists, but normally stalks are vertical and you can distinguish it by absolutely other characters. So probably it would be a good idea to wait several years until more specimens would be collected. And then we would have no need to amend the description of the species. However, okay, now we have 2 papers instead of 1. So I support this idea. As for the locality, we cannot exclude the endemism, but if you find something on the same log year after year, it can be, for example, an apomictic population which was formed there and which will never form a real big population. And one day after a strong winter, it may just disappear, and that's also. Such cases should probably not be described as separate taxa.
So the locality should mean different forest pockets, different logs, something.
At least different logs.
I'm strongly opposed against ad interim names. I know from my life in the mycological societies that they can start leading their own life, and they are quickly accepted as really described species among people, and you find them all the time. If you want to publish an undescribed species, just put it as SP. So I would not give it a name or give it a number code, but not a name, because that will lead to a lot of confusion.
All these should be very careful with the name, or whatever getting out. What if we just use the vernacular name? It's a kind of nickname for it.
I have a comment to make on the name, and then also whether you want to have I think you suggested publishing some information in the slime mold journal if you find something, hoping that somebody else will find some more material. I actually think that's quite a good idea, and that's how I realized that David Mitchell had found what I had found previously. So in one of his papers, he had written that he had discovered something in Australia, and it just so happened that I remembered reading that previously. So then I knew that there was a collection somewhere else in England, and that I could then proceed because we had 2 separate populations. The other thing about the name is I don't remember what he called it at that point, but in Western Australia, because we find new native species all the time, not slime molds, just vascular plants, we needed a system to be able to name those plants so that we could get them onto the rare and priority listing. So it would be called, a species would be called something like Lamproderma sp brown peridium. So it would just be a descriptive term and could never then be converted into a name that people would consider to be a named species. And that has worked exceedingly well because that name goes on our census. And then over time, when that eventually gets named, you just link that as a type of a type of synonym. So anyway, there are ways around all of these things. But were you suggesting that if you find a new species and you were hoping that somebody else found it, that you would just put a small publication in the Slime Mold Journal? Is that what you were suggesting? Yeah. Okay. Cool.
Would that be a solution simply to use a really descriptive name? So that's going to be a mix of this program with the abilities.
I think we have two examples already in Myxomycetes. One species was Lamproderma cacographicum that was described as Lamproderma sp and then afterwards discovered by Marianne on description that she has the same specimen and she described it based on another material. And the second is Meriderma. Three species are still recognized before they are described. Yeah, this a few species we know use the names, which are descriptive names, which is very helpful in my opinion, but they are not yet described. So I think that both examples can work well. It is just the way of communication, and my personal opinion is that it is more helpful if we have a descriptive preliminary name or at least a description than simply species, I mean SP, because we can have Diderma SP1, SP2, SP2, and when you find something strange, how you find which of these SP1, 2, 3 you have. So yeah, like that. And also, if I can continue, I would not vote for around 3 collections. I would simply say more than one. This is the general practice in taxonomy, also in mycology, that single collection is not enough, but more than one. So it is even put in the instructions for authors in mycology that you cannot publish a species if you have only one collection. Yeah. So I wouldn't say that 3 is better than 2 or 4 is better than 3, but just more than one. And of course, it depends on the situation. And sometimes you can, if you are not sure that the second collection is really the proof that you have a good species, it's better to wait for the 4th one. So it's for decision of the author. And also, as you can see, the examples of Lepidoderma stipitatum and Crassipes, it was described from one collection or maybe 2, and it was also found on another collection in another part of the Europe, and it was the same, and it turned out to be synonymous with other species. So I think the Myxomycetes are very tricky.
Okay. Yeah, so I have not enough time to change it on the slides, but I simply recorded more than one collection and use descriptive things. That was the conclusion.
Just a remark. In general, I agree with this point, but sometimes there are cases where we can come across something really, really rare, and this would be the single collection, but as in the case of Spiramyxa that I showed in my presentation, there was a collection that is documented from the plasmodium through all the stage of ripening, the maturation, and it is really a rich specimen. And it has really unique phylogeny and unique in many ways, many more morphological aspects. And there is no other collection and nothing I ever seen even like similar to that. So in this case, this really interesting genus, we should just call what?
I would like to remind you of the leaves I passed around from the trees. what is if you have this first all of the landscape leaves, and I'm sure it has been described something different in the early times. It is super distinct, but on the end, you see that the distinctiveness is based basically on a single character in the few genes. I would recommend these super distinct cases. You then should have a molecular description, and you should make sure that your unique characters are actually a combination.
Yeah, exactly. That's the case with Spiramixa, and with 2 genes sequenced, I have like zero doubts that this is something very special.
So there may be exceptions, but then I would say we need molecular group and a combination of characters. Okay, just we are a little bit short in time, so I proceed to the next recommendation, which is probably a relatively simple one, comprehensive. That is, of course, the best what we can do to leave for our successors. So here's a to-do list, what we could do. What I sometimes do is geographic coordinates to make the new entity. So worldwide databases right now, everybody can record geographic coordinates, another big plus for citizen science. Every smartphone, of course, will do. So here's a to-do list. We have okay. So the exception would be if you describe in the Hawaiian space. Yeah. I quickly recall these things in my notebook, so still we have the idea to do a little publication in Sligo Journal about this. Yeah, of course. If you discover in Hawaii, when all your own Hawaiian specimens have no geographic coordinates, you can reconstruct with the students you can.
Just for this one, it's very good to document the morphology in subtle people or in fresh state, and if possible after, if something changed after several months of preservation.
Yeah, that of course is very understandable when it comes to the problem with Arcelia. The Arcelia occidentalis may fade even fraction. Just sunlight plus oxygen, the compounds are, I think, nothing enormous trying to form right, and they quickly degrade. Yes. More additions. I just recall. Then process rule number 3. Oh, that was only the illustration from comprehensive description, not needed anymore, of course. Rule number 3, find diagnostic characters. And here, my rule includes more a disclaimer than anything else, since I hadn't have the time to write this down, but with spores, you should be cautious. The environmental cause variation in spore size is really, really large. The quantitative measurements for spores have exactly the risk of that. So we should be careful in finding diagnostic characters. And that's the problem I would like to ask you. What species should be included for comparison? Only the morphologically similar species, or also the molecularly similar species, which is the difference.
I would say that the first of all, we should compare with morphological species because we still follow morphological species concept. And then I would also like to comment on quantitative characters, that sometimes the characters distinguishing the species are not quantitative. For instance, Niviculus didermas are the most easily recognized macroscopically on some macroscopic characters. They have spores almost the same. So spores don't differentiate them.
Yeah, the point is, of course, that we should not limit ourselves to quantitative character.
I think we need find morphological differences in genetic like species. For example, we have a lot of species, twin species, like it's not molds, but I have one example. It's Calmusia varispora and Calmusia longispora. They genetic sequences is very similar, and it's difficult to distinguish it with genetic study, but pretty easy to distinguish by their morphologic.
I think that's a good thing. Molecularly similar species need to show and we need to document morphology.
I was just going to say, in my prehistoric education, we were taught that species should differ in at least two independent morphological characters. You stated here at least one. Just a comment.
I also like to see that the exchange just at least two different characters to be independent.
In the last discussion, in the last day of the Congress, we will discuss what to do with the cryptic species. And actually, if we recognize the biological species concept, we can imagine the case where two real species do not have two morphological differences. This is possible. No one can come to Myxomycetes and say you must differ from each other by eye-catching morphological characters. So twin species exist, they may exist. So if you don't have nice quantitative morphological characters, this doesn't mean that you cannot describe taxon. It means only that the requirements to the molecular proofs of the novelty are much stronger. But if you obtain these molecular proofs, you still can describe a taxon even without prominent morphological differences. I would say we cannot forbid this. Because
yeah, but I think it did not mean exclusively morphological characters. That can also be, for example, pronounced difference in ecology.
That's true.
Just a theoretical, hypothetical question. I know that color of plasmodia, the pigments, can be important. Would you describe a new species if you saw two different colored plasmodia producing exactly identical sporocarps?
Clearly, no, there was
Yeah, exactly.
And he, if I recall right, he was saying that the plasmodia can take up something from the environment and that changes the plasmodial color. But if you see in the field, in different specimens, the same difference in plasmodial color, Tasmanian myxa, the plasmodium is this pale sulfur yellow and not white like in other Lamproderma, sure, that's the point. So yeah, be careful with that. So I make the recommendations and elaborate that and maybe we can pass the manuscript a little bit around. We have to see it in middle how we do it. Number 4, that I think is a good suggestion. Obtain a barcode from at least a single genetic marker for the new species. Best sequence it for 2 or more independently inherited markers. But the molecular barcode, whenever possible, this is really important. This will allow to prove the accessions of the new taxon to form indeed a monophyletic group. That's also important. So having more markers increases the resolution, of course, clear of phylogenetic analysis. So if possible, a molecular signature should at least be attempted, I think. And that is nowadays, I think, possible, since also when you are a non-institutional researcher, you probably find one of us institutional researchers which is willing to do the molecular work, at least to obtain a molecular signature. I would always try to volunteer in some cases.
These are actually several requirements, 3. One stronger than another. The first requirement is a presence of a barcode itself, at least for one gene, and I would say this is obligatory now. It is strongly not recommended to describe taxa without molecular barcodes, which show the molecular uniqueness of the species of the taxon. The second requirement is that it must form a monophyletic branch and do not branch within known taxon. Yeah, exactly. But well, if it is, if you have only one accession which has several unique mutations, but it still branches within another species, this also cannot be recognized as a new taxon. So these are 2 different and both important requirements. And of course, if we have at least 2 markers, we can check a reproductive isolation. It is the strongest proof of the novelty.
I would like to add a little bit to the criterion of a new species forming a monophyletic group, not included in in a clade containing other species. If you have a single gene phylogeny, it is possible that a real good biological species can form a polyphyletic group or be paraphyletic, because genes, evolutionary history of genes can have very difficult, complicated pathways, complicated history, and due to incomplete lineage sorting or horizontal gene transfer, you can observe in a single locus tree the non-monophyletic groups formed by a really good species. So if you observe a non-monophyletic group of a species that you think is really a good species, then it is, I would say, obligatory to study other genetic markers to confirm that it is indeed a monophyletic species. So if you have just a barcode sequence and it doesn't show what you expect to see, just try other genes.
Yeah, exactly. Moreover,
where we have just a branch within another species, and the branch was divided.
And if you have big conspecific branch made of sequences retrieved from gene bank, and your species is within it, it doesn't mean this whole branch really represent one species. It can be much more complicated, of course. But this requirement still can be stated, because if we have such a case, it means that you need more studies to prove that your taxon is new. So this is possible, that you find your species within the branch of other species. This doesn't close your hypothesis completely, but it means that you need more data to support the separation of the new taxon.
Just one remark that I wouldn't say that it is obligatory to sample, I mean, to get sequence from the new species. And I don't know if, for instance, Gabriel Moreno, who is now going to describe two new comatrihas, he doesn't include molecular phylogeny there. So I would say that it should be still possible to describe a new species because it is not obligatory by the code to include sequence. And some specimens have such a nice morphological characters that we can check it later. Yeah. So verify later the position. Of course.
In that case, I think we could do specimens, species, we could at least do part.
But you probably don't have enough comparative sequences of comatriha, so you cannot do it without it.
Yeah, that's true, but having a molecular signature which is connected to the type specimen will help our success of scraping, even if you don't have any material from them.
No, but when you have other described comatrihas and you don't have sequences from them, you are not sure if you have already described species or not.
Of course.
Yeah.
But I think the sensible recommendation is that immediately try to link a barcode to a typus, to a holotype. And that could be done regardless if you have comparison material or not. So it's difficult for Martha, I absolutely agree, but at least we should in this case make a serious attempt to do a barcode with Martha Adina first. So we might have two chances that works with the other as well. And you always should try, and that is in my case a good recommendation, to find a partner which can try it for you. Of course, you might have the species, look at the Bugala, what was it, this could be last year, where it is super. But at least it would help to recommend that here firmly, you should try the same part. That's right. This is another step. For example, if you describe scope in many cases, it's not really diagnosed. But you still describe this character. And as well, you can describe SSU gene, not to distinguish it immediately from the rest of the species, but just to have this information. Yeah, so I think this is recommendation. The first column, independent from the point, if you have comparison sequence go up, is science makes progress and eventually you will have progress. And the number 5, molecular differences to a class search. There are two interesting papers by Kirchner et al. Regarding ascomycetes. As an example, one paper was just coming up with rules for a class search. It's quite nice, described also for students, and the other paper is just exercising this on the example of a newly described classification. But that would also apply for our case quite nicely. So if you have a barcode, do a class search and try to see what are your molecular close species. We will tomorrow probably from the arena hear more about this and the tricks and difficulties comparing it, right?
Yeah. After tomorrow, yeah.
So the day after tomorrow, we will hear more on that. But I think that could be a good recommendation. Sure, you could and here the recommendation
because it proves that your species are not known.
It's pretty good. Let's search and it says DD and this.
Wait, where's the thematic tube?
Yeah, that's designed for it. Just a little remark. The last Congress in Costa Rica, I remember there was a discussion about the reference database on the barcodes for Mixos. Yeah, I understand the problems that are behind that, but it would be very useful to have a reference database where the Mixo people could blast their barcodes across the reference species. We really lack this.
Yes, our Russian colleagues have started with Vladimir Tmoshinsky and Andrei Matveyev. But of course, now we have this unfortunate political situation. I have to ask them, nevertheless, it would be nice if we could also use this as a databasing attempt for the whole community, since databases make only sense if we can use them all. I'm completely agreeing with it. That would be very nice. We have this database already up and running. It is working, I think, Oleg, but yeah, we have to make it publicity available. Yeah, that's a good point.
Yeah, I also agree that it would be super useful for all of us. So far, we just didn't really put enough effort into creating it, into maintaining it, into making it easy to use with easily understandable structure and so on. So yeah, so far we're using it mostly for our internal data storage, but it's of course not how it should be. Yeah, sorry for that. And I would like to add a little bit to comment on the recommendation to ensure that the sequence difference is above the barcode gap for the respective group. I think it's a very tricky recommendation because barcode gap, yeah, maybe Irina will tell something much more detailed on that. I'm really looking forward to her talk. But I would say that barcode gap is a very artificial construct, and we shouldn't constrain ourselves to some artificial thresholds of barcode gap of how different should be two barcodes from each other to say that species is indeed a different species. We observed many times that two really well-defined species can differ in just one or two substitutions in the barcode sequence. And yeah, it doesn't make them bad species. So yeah, that's it.
Noted, yeah. So we cannot put here in a certain recommendation, you should have a minimum threshold for a barcode gap. And I think, Irina, this is also what you want to say, basically, right? Yeah, okay, it's noted and will be changed, of course. Regarding this sequence database, yes, I was a little bit, to be honest with you, counting on it that we find the joint funding line, but the outbreak of the war has all disrupted this. All the funding lines together with Russia were immediately suspended, so we couldn't find any more funding, and that's of course tragic. So we have to see what would be a solution for that.
And a small remark from me about the blast search, it can help only to some extent to be at least sure that your sequence is definitely something unique. But unfortunately, blast search can't answer all your questions because I remember from my moist chambers that my favorite story when I put my sequence in blast search, and the one thing he found, it's uncultured soil eukaryote number something from a metabarcoding data. So yeah, sometimes it really can't help.
Yes, be aware that the thing which comes out from BLAST might be a wrongly annotated sequence. Exactly. That always is a point. Here it is. So number 5 was blast search. Number 6, name your species carefully. I would say, and that was my reading from the article of Roniker and Alansky, which explained why we should right now stick with a botanical code of known flesh. You never know what the future will bring. Maybe there will in future be an attempt saying, okay, now we need to go according to a natural system also with the divisions, what is zoological code, what is botanical code. So it might be a good idea, for example, not to create a genus if we have a new genus, which is used already in the realm of zoological. At least not to make the confusion more. So don't use a genus which is used already by the zoologists, and that should be easy to do. And I would also not recommend to use an epithet which is already in use for any other mycelium species, especially in a related group. Yes. So this would be also two recommendations, this number 6. So the first is no epithet what is used in myxomycetes, especially in a related group. And the second is not using a generic name which is used in zoological code. Any additions, any remarks?
One addition, right? Not using the same epithet. I would say not using the same epithet is a bit strict, no? Because there can be many hundreds more species, and if you use vulgaris once, you may use it once again. So it's a bit strict, I would say, even though I'm not a mixomethodologist, but as a taxonomist, I would say it's a bit too much.
Yeah, I think we still can manage with some 1,700 names. I would say, okay, let's say not use an epithet in use already in any recognized myxomycete species. And that should be very easy to do with a simple search in Carlos Nomenmux database. Maybe rephrasing that, not use an epithet which is already used in a recognized species. And now we are soon at the end, since I have trouble with times. That should be clear. Deficit closure specimens in a way that these can be used for further study. I came across an interesting paper in ferns, and they checked the viability of DNA in fern spores and found that if you store it at lower temperature, it greatly prolongs the viability of the spores. They wanted to do as a first target flow cytometry with the spores, but I would say that applies also for our programs where we do first order sequencing with the spores. Might be a good idea to conserve a part of such a type specimen in a fridge. Interesting, at least an idea. But depositing a vulture specimen, and maybe if possible in several herbaria, would be a nice thing, since we still have too many cases in myxomycetes, C. leucogala, where we cannot access later a specimen. What was it? Confusum was the problem. Leucogala confusum. Such things we should avoid in future.
Just related with the 4 degrees Celsius, actually, as I showed, the parasites sometimes germinate within the 4 degrees Celsius. So if you treat about the Lamproderma with the not completely dried, it might affect, degrade the myxomyces spore itself. So you should be careful about the degrees and also the moistureness, including the spores. I mean,
No, not deep freezing, just 4 degrees and dry.
Yeah, completely dry is quite recommended. Otherwise, some of the other molds coming to that one. Yeah.
4 degrees and dry. I will add this as well. And now I'm on the end of the small discussion regarding my recommendations. Any more to add? Would we recommend voucher in several public herbaria? I think that is a good thing. Of course, at least one, yeah. Drawings. Drawings are nice, but as an add-on, I would say. Drawings are always nice. Of course, I always tell the students when we compare photographs and drawings have three advantages. If you are a really experienced researcher, in a drawing you can show the truth. In a photograph, you always show certain specimen with all its typical and even non-typical characters, but in the drawing you can highlight typical things. So drawing is a good recommendation, at least for somebody who is as good in it as me. That's the point. Any more?
One. Only the scanning electron light.
Yeah.
It's nice to talk about the. It's also useful for the.
Yes, yes. You should, that is a very good recommendation, not only scanning electron microscopy, but also things which you can easily comprehend as a non-institutional researcher. And especially nowadays, the stacked photography, which is done by many non-institutional researchers, opened wonderful opportunities really to document special species.
Which remind me about one thing that I often notice in taxonomic papers overall, regardless of the topic, is the absence of raw measurements, because we always see the condensed measurements, like from which value to which value spores are. But what I'm really missing is like the raw table of like each spore, because this will make data reproducible, and this from this data you can do some more analysis and to see some more interesting things, to see things overall, several groups, etc. So I think this is a thing that taxonomy really needs overall more.
So you're advocating for using, for adding the raw spore measurement data as a supplement, for example?
Or any kind of raw data on measurements.
Yeah. Yeah,
Yeah.
never hurts, you know.
Yeah.
I would say. Any more recommendations? I'm ready to throw the cube.
I have a question regarding your question or your comment, because only for spores or for everything? Because I mean, if you need to measure and to note every, I don't know, the size of the sporocarp or the diameter or whatever, it's super time consuming. Yeah, we do it, but do you write it down and give it as a supplement? That's the point. That is more, I don't know, more tedious, let's say. Yeah. Mm-hmm. Yeah, okay. Yeah, I see.
Yeah, but at least for such a difficult thing like the spores, it's a good recommendation, and that is really quickly done. Simple.
Yeah, with the spores, yeah.
And recommend and note if you measure it with or without ornaments.
Mm-hmm. Where do I throw or no?
So more like a question, because of, as you have talked about the spore size in particularly Nibiculus myxomyces dark spore vary a lot. So when you measure spores, I tend to try to ignore the abnormally large ones if there is great variation. So is there a recommendation of what spores to measure? You get a gut feeling after a while what are the typical ones. So you try to avoid the abnormal ones that could be abnormal for various reasons, but just a point.
Yes, very good point.
it down, think about the second spore population in one of these specimens of I have shown for ICSEM11. If you measure it all strict automatically, you will end up with a very wide range. If you look carefully as an experienced researcher, you realize, okay, there's an operation in this particular specimen. It has so-called markers.
The raw values can easily be checked for outliers, and this is what can be recommended.
Yes, check for outliers. Yeah. So thanks a lot for all recommendations or more recommendations. I'm always happy to take, but at least right now we have together already a primer for such a publication in Science, and maybe we can do this for the other discussion rounds.
I think we end up with quite many recommendations, so we will have the most strict rules of description of species ever. I mean, so I would simply suggest to divide to the recommendation and maybe suggestions, because if we put all together as recommendation, then nobody of us would like to describe a new species, I think. The most important and really necessary as recommendation, and other are suggestions, because I think that we can accept description of species without, for instance, these measurements, and some if it is not necessary, and so on and so on. Yeah.
Yeah, so we should denote our points in two categories, recommendations, and suggestions. No. And of course, any more things we do the most is already. But the bus is coming at right now, so we already successfully did the idea. But in any case, we have now the story. But I'm very, very happy for anything which later on I will and I will try to make my market for it in this one. You have time, you have time.
U 4 chyba właśnie nie skończyli teraz, tak? Nie będzie posteru.
Okay, go to the books.
We go to the clusters, and if something in restaurants, square time in between clusters, and in general,